Okay, now that we have talked about what happens in the dark, and we've said these rods or photoreceptors are depolarized during the dark, and they've got about the memory potential of about negative 40 millivolts. Now, let's see what happens when now light rays have been shown into the eye and they've been absorbed by the retina. Now, what happens, what really takes place for action potentials to be generated was we're saying we're changing light energy into receptor potentials that are generated as action potentials and then sent to the visual pathway. Now, we'll go back to this diagram and we've said this one here is rhodopsin, the photosensitive compound rhodopsin, coupled to transducin, a G protein with the alpha, beta, and gamma subunit. Then this one is a phosphodiesterase enzyme. This one is a guanine cyclase enzyme. And I said this one converts GTP to cyclic guanosine monophosphate, which in the dark increases and binds onto these cyclic guanosine monophosphate gated channels, thereby allowing this sodium that leaked out from the inner segment to go back into the cell through the outer segment and through this channel there, as then calcium channels enter while these potassium ions are leaking out through these non-gated channels, depolarizing the cell, leading to the release of neurotransmitters on the bipolar cells. The glutamate inhibits these bipolar cells. Less or no release of glutamate happens on these ganglionic cells, and therefore no action potentials are generated. That's what happens in the dark. Now, when light rays have been shown here, what they will do is they will hit this rhodopsin. So these light rays will hit that rhodopsin. And what happens that rhodopsin is made up of scotopsin and retinal in the form of 11-cis retinal. Now, when light rays hit this, what they will do is they will convert this 11-cis retinal into all-trans retinal. And from the beginning, I said that only 11-cis retinal has that confirmation that is needed, to bind to scotopsin in order to form rhodopsin. So because now the conformation has changed, it's now an all-trans retinal. It's no longer 11-cis retinal. What will happen is that this scotopsin will move away, and this retinal, 11-cis all-trans retinal, also moves away. So if this was rhodopsin, scotopsin here, and this one was 11-cis retinal at the bottom, what happens that they now separate like that. Then you have all trans retinal here. Then you have scotopsin there like that. Now this scotopsin, separation of all trans retinal from scotopsin causes activation of this transducin there, the G coupled protein. This gets activated. When it gets activated, it converts GTP. So transducin there, when it gets activated, it converts GTP into GDP like that. Then this alpha subunit, let me use my green marker there. This alpha subunit moves away together with the phosphorylated like that. It's now active. What it does now, it goes and activates this enzyme, phosphodiesterase. So let me show it properly here. When that separation has happened, this separation causes activation. So this is scotopsin and rhodopsin. They have separated into scotopsin and all-trans retinal. Scotopsin I'll put SP, then all-trans retinal I'll put A, T, and R, all trans-retinal, have separated. This separation causes activation of this G protein that is coupled to rhodopsin. We have got the gamma, beta, and alpha subunit. The activation of this G protein causes the conversion of GTP to guanosine diphosphate, GDP, like that. Then, due to this one separates, the alpha subunit separates from this, the rest of the subunits, the gamma and the beta subunit, and it moves out. Since it has now been phosphorylated like that, it moves out with its phosphate group like that. When it moves out to the phosphate groups, it goes and activates phosphodiesterase enzyme. When this phosphodiesterase enzyme is activated, what it does is that it will convert, this enzyme will convert cyclic guanosine monophosphate to 5' guanosine monophosphate. It converts 5 cyclic guanosine monophosphate to 5' guanosine monophosphate. So in other words, when this enzyme has been activated and converts that, it gets to reduce the concentration of cyclic guanosine monophosphate inside this cell. Since CGMP levels go down, as the levels of 5' guanosine monophosphate go up, this 5' guanosine monophosphate cannot bind the channels. Which channels are we talking about? This 5' guanosine monophosphate cannot bind here. It cannot bind these channels due to the fact that this cyclic guanosine monophosphate won't be available now. These channels will do what? Will close, as shown there. They will close like that. When these channels close, remember, during the dark, there's sodium moving out and moving in like that through those channels, and there's calcium moving in, there's potassium moving out through those channels. Now, these channels have closed now. Since there's no cyclic guanosine monophosphate to bind here, the channels have closed. Now that they have closed, sodium cannot enter as shown by that arrow. Potassium, calcium can't enter as well. So you can't have influx of calcium, of sodium, and influx of calcium like that. But you do have efflux of potassium there. So since there's more sodium, the sodium is being released out, and more sodium is being released outside there, these cells now will hyperpolarize. When these rods hyperpolarize, these channels that are present here, remember the channels at the synaptic terminal, the voltage gated calcium channels, less of them are going to open. Less of these channels are going to open. So for example, let's say, just an example, let's say we have 5 of them. Maybe one will just open. One or not, or all of them cannot open. So now, since one of them has opened, or less calcium channels have opened, less calcium enters the cell. Less calcium enters the cell, there are influxes there. Since there's less calcium entering there, less glutamate is released here. So there's less glutamate released here at the synapse between the rods and the bipolar cells. Since there's less glutamate here being released, and glutamate on the bipolar cells is inhibitory in nature, since there's that less inhibition, the bipolar cells will become excited, or will be activated. Since they become excited, these will release more glutamate. They release more glutamate on the ganglionic cells. And since there's more glutamate released on the ganglionic cells, these ganglionic cells will become stimulated or activated, and an action potential is generated. When that happens, when that action potential has been generated, we're now going to say that phototransduction has occurred, because light rays that were hitting this rod here, like we have shown here, has been converted into an action potential. Okay, let's run through what we are saying here. What we are saying is light rays have hit this rhodopsin there in the membranous disc outer segment of this rod there. When that happens, after hitting that, it gets converted, since rhodopsin is made up of scotopsin and 11-cis retinal. Since when light rays hit this rhodopsin, this scotopsin gets to move away from the cis transretinal, because this 11-cis retinal is converted to O transretinal, and this O transretinal has the conformation that is not going to fit with this scotopsin to make rhodopsin, so they dissociate. This dissociation causes activation of this transducin this G protein, G stimulated protein. Its activation will now cause the conversion of GTP into GDP. Then this alpha subunit will move away with the phosphorylated. There you can see that GTP has been converted to GTP. One phosphate group remains. The phosphate there goes together with the alpha subunit. This alpha subunit is going to go and activate this enzyme, phosphodiesterase enzyme. When this phosphodiesterase enzyme is activated, it gets to convert this cyclic guanosine monophosphate, which during the dark was increasing in concentration because GTP was being converted to cyclic guanosine monophosphate by this enzyme guanylate cyclase. When this phosphodiesterase enzyme is activated, it gets to convert this cyclic guanosine monophosphate into 5' guanosine monophosphate. This 5' guanosine monophosphate, unlike CGMP, cyclic guanosine monophosphate, which can bind onto these channels there and allow this sodium that was leaking out to move back into the outer segment and again allow calcium to move in. This 5' guanosine monophosphate cannot bind here. Due to the fact that it cannot bind here, these channels close. When these channels close, no or less sodium and calcium enters the cell. And since no or less calcium enters the cell, as well as more potassium is moving out, because this channel is then gated, it's just leaking out potassium channels. More potassium moves out, less sodium is being moved, more sodium is being moved out as well, and less is being gotten in. This cell now hyperpolarizes, and because it hyperpolarizes, there is no that stimulus that can activate these voltage-gated calcium channels. Since less or no voltage-gated calcium channels are being opened up here, less or no calcium enters the synaptic terminal. Since less or no calcium enters the synaptic terminal, less or no glutamate is released here on this synapse. Where is this synapse? This synapse is between the rod and the bipolar cells. Since less glutamate, which is inhibitory on these bipolar cells, is being released, these bipolar cells, their activity increases, or they get much more stimulated or excited. Since they are much more stimulated or excited, more glutamate is released by these bipolar cells onto the ganglionic cells. This glutamate released is excitatory on the ganglionic cells. So it gets to stimulate these ganglionic cells, and therefore an action potential is generated in these ganglionic cells. Because at the beginning we said the ganglionic cells, unlike these bipolar cells and rods, these ganglionic cells get to generate action potentials. These two generate receptor potentials. When that happens, we say that action potentials have been generated, or phototransduction has occurred. So if someone asks you to talk about phototransduction, what you talk about the scenario in the dark, then you talk about the scenario during the light. You talk about the closure of these channels as well as the influx and efflux of the ions. There's actually nothing much confusing here. All you have to do is just take a look at this video, then go and do your studies, go into books, as well as couple this video with other videos. So with that said, we are done with phototransduction. Don't forget to subscribe, share, comment, like, and suggest the topic that you want.